Okay, so let's hope I get this right this time. Last time through, I didn't have my microphone in the correct position, and consequently, the audio didn't record. I know that's happened before, hasn't it? Well, anyways, it was a while ago that I recorded the original version of this, and so I'm just hoping that I can remember what I was supposed to say here. I know that I know that I started with a review of the chromosome structure, the genome structure, right? We said that the entire genome of eukaryotes is so huge that it makes sense for it to be organized into particular sections. We call them chromosomes. Each chromosome consisted of a single linear piece of DNA. It's still a double-stranded molecule, right? Because DNA is always double-stranded. But we only have one double-stranded molecule of DNA, which corresponds to one chromosome, right? Moreover, the chromosome, the DNA in a chromosome for eukaryote, at least, was packaged into a material called chromatin, which basically complexes the DNA with the histone proteins. Now, the other thing that we went through, and I want to show you this other diagram, which I tried to recreate what I did in that previous video, okay, was to say, well, at some stages in the cell cycle, specifically in G2, right? Because G2 is after the DNA replication takes place. During the S phase of interphase, the DNA gets replicated, and so from the end of S phase through G2, the chromosomes consist of two sister chromatids, right? And so these two would be sister chromatids. But another thing about the way that I drew the chromosomes in that diagram is I tried to be relatively accurate in the sense that it's putting together relatively matching sets, matching pairs of chromosomes. So if you look back at the previous video, there would have been two chromosomes that are about the same size, had the centromere in about the same location, and these corresponded to two homologous chromosomes. Which basically means we've got two different versions of each chromosome in our cells, because we are diploid organisms. We get one set of genetic instructions from mom, and so maybe this was a chromosome that we got from mom. This would be the chromosome that we got from dad. And so we can actually label the chromosomes according to whether they're maternal or paternal. And if we just number the chromosomes, this might be chromosome pair number 1, this might be chromosome pair number 2, this might be chromosome pair number 3. And so as such, so this chromosome here would be M, maternal chromosome 1. This one over here, you'd say that one is paternal chromosome 1. This would be, maybe this one is paternal chromosome 2. And then this one would be maternal chromosome 2, because we've got two chromosomes too. One of them has to be maternal, the other one has to be paternal. And maybe for chromosome 3, we'll say this one is maternal chromosome 3, and this one would be paternal chromosome 3. And so we're pretending that this would be an organism, a eukaryotic organism, whose genome consists of 3 chromosomes, right? So if this were a human genome, you would have 46 total chromosomes, but there would be 23 different kinds of chromosomes, 23 homologous pairs of chromosomes. So you'd have M1, P1, all the way to M22, P22. And for that 23rd pair of chromosomes, you would have the sex chromosomes, either X or Y. Okay, and so one concept that you have read about at this point is that we've got another element to genome structure, in the sense that our chromosomes are organized in as specific sets of information in which you're going to be inheriting one copy from each parent, right? And so one thing you don't expect, you would expect the genes on the two chromatids to be the same, right? So if we have a gene over here for eye color, gene on this side for eye color, they should be the same because they were produced by DNA replication. Okay? There's no reason why one sister chromatid would be any different from its sister because they were produced from just copying the information that was there in the original chromosome, right? But if you look at paternal chromosome 1, the genes that you have on paternal chromosome 1, yeah, well, they should be the same, right, between the two sister chromatids, but we don't necessarily expect there to be concordance between the two homologous chromosomes, right? Not. I mean, they could be the same, so not necessarily the same, because after all, we're getting different sets of genetic instructions from each parent. And so, if this is the gene that we're inheriting from mom, we might be getting a different eye color gene from dad, right? And that's the nature of homologous chromosomes. It would be the same information. They would both be genes for eye color. Well, it doesn't have to be eye color. It could be something else. It could be coat color in the case of a mouse. But the genes on either sister chromatid would be the same, but the genes between the two homologous chromosomes would not be the same, right? And the same goes for all the other pairs are the same. You would expect the same to occur on each sister chromatid, but not necessarily on the two homologous, on the same position of the homologous pairs, right? Got that sunk in? Okay. And so this kind of sets us up with the ability now to start talking about meiosis and the process of recombination and how we're getting brand new combinations of genes as a result of that special kind of cell division that occurs that leads up to the production of gametes, right? And so let's look back at our outline. We have reviewed the structure of the genome, pretty happy with that. We have the concept of homologous pairs and how what this means. And so homologous pairs of chromosomes are potentially different sets of instructions. Now, they don't have to be the same. You could have gotten the same gene from mom and from dad. And that's totally a possibility, but there's no requirement, there's no expectation that the genes are going to be the same from the two parents, right? Now, I mentioned this briefly, but diploid refers to the chromosome number. Di is the Greek prefix for 2, right? And so a diploid genome would be the state of having 2 haploid genomes. The haploid genome would be the one full set of instructions coming from one parent, right? And so the reason why diploid organisms have 2 sets of genetic instructions, because we're getting one complete set of instructions, one complete set of chromosomes from mom, and one complete set of chromosomes from dad. And in the case of humans, you would say the diploid number, the 2n, is equal to 46, because you would include both the autosomes, the 22 pairs of homologous autosomes that don't have any role in sex determination, and the X and the X, or the X and the Y, depending on whether you're a boy or a girl. Right? Haploid refers to the state of having only one set of genetic instructions. And so if you look at human gametes, sperm or eggs, each sperm carries only one full set of genetic instructions. It has to fuse with a full set of genetic instructions, the haploid set of chromosomes that are there in the egg. And when they fuse together, we call that process fertilization. And we end up with a nucleus with 2 complete sets of chromosomes. In other words, the gametes are haploid. You would say the haploid number for humans is 23. And gametes, sperm and the eggs, would be the entities in the life cycle of humans that are haploid. Whereas once you have fertilization occurring, the fertilized egg, that zygote, and all the cells that come after the zygote, right? fertilized egg occurs. The fertilized, that would be the zygote. Then you would have a cell division. You have another cell division, so you'd have 4 cells. Then you would have 8 cells happening. Then you would have 16 cells and so forth. Basically, you keep on going, cell division after cell division, and these are mitotic cell divisions. Okay, and with mitosis, we're not changing the chromosome number, and so once you get to the diploid state, mitosis is going to give rise to, 10 trillion cells, right? 10 would be 10 million, that would be 10 billion. This would be 10 trillion cells in the adult human body that are all descending through mitosis from that single zygote, and they will all have the same chromosome number, right? And so you would say, well, the entire lifespan of your typical human would require that every cell in the human body would have 46 chromosomes, and the only exceptions to that would be the gametes, the sperm and the eggs that you could be passing on to your offspring, or I guess the cells immediately preceding the gametes, because before you have a complete sperm cell, you have another sperm cell, spermatocyte, that has undergone meiosis and has not yet completed. It has not become a completed sperm cell. But yeah, there's a very limited number of cells that are not diploid in a human body, and those are the ones that are associated with reproduction. Right? So pretty clear on what a gamete is. A zygote is the fertilized egg, and basically the entirety of the human existence, we're thinking about diploid cells. Fertilization is the process by which we get a diploid set of chromosomes from the 2 haploid sets of chromosomes, one in the sperm, one in the egg, that come together and fuse in the nucleus of the zygote. Okay. Okay, now I guess what we're describing here with fertilization, I got make that a T, make this an L, fertilization. Okay, coupled with meiosis. Okay, meiosis is kind of like reduction division, where a 2N cell becomes a haploid cell, and fertilization is the fusion where 2 haploid cells become a diploid cell, right? The combination of these 2 things is what constitutes sexual reproduction. Okay, and these are 2 things that are, if you notice, they're completely absent in asexual reproduction, which is something that we talked about in a previous video. Remember, we said that like a starfish, you break a leg off of a starfish, right? Break this leg off, and eventually that leg grows into its own little starfish, right? Remember that picture? And yeah, you could do the same thing. Basically, if this happens, the offspring has never undergone meiosis. It's never undergone fertilization. This would be a completely asexual process, right? So yeah, I mean, with asexual reproduction, you can also say asexual reproduction is ameiotic, without meiosis. Okay, ameiotic, it's an ameiotic process. Okay, so the thing about sexual reproduction is that it gives rise to a genetic diversity, which is going to be the key concept. It's kind of like the key reason, right? It's one thing to know what sexual reproduction is. It's a much more important thing to know why sexual reproduction is there as a common feature in the life cycles of most every animal and plant that you come across, right? Why do organisms continually to reproduce sexually? Well, a big part of that answer would come down to the amount of genetic diversity that occurs as a result of sexual reproduction, right? Now one form of that genetic diversity is something that you might be thinking about here, right? Because we have genes from 2 parents, we actually have different kinds of genes, right? We might have a gene for pink eyes and a gene for black eyes within our own genome, because we're actually getting information from 2 different parents. So that would constitute some amount of genetic diversity, right? But really, that's not the genetic diversity that we care about. The more important kind of genetic diversity is the kind that comes as a result of sexual reproduction and the amount of genetic diversity between individuals in a population, between individuals in a species, even siblings, even like brother and sister. Right? You can think about this even before we get into the discussion. A brother and sister, they both share the same parents, right? They both come from the same mom and the same dad, and so they must have the same genetic information, right? Well, no, you know that's not true. You know that most full siblings, even though they share the same parents, they're not identical to each other. And the reason to understand it is it's perfectly obvious, right? The brother might have inherited the gene for pink eyes from mom, and the sister might have inherited the gene for black eyes from mom, right? Because if mom has this genotype here, where it's got one gene for pink eyes, one gene for black eyes, then one offspring could be inheriting that copy. The other offspring could be inheriting that copy. It could be that both offspring are inheriting the same copy. That could happen as well. But the idea is that when the gamete is formed, when mom produces an egg, we have meiosis taking place, giving rise to an egg. We're getting a combination, a kind of a random combination. I'll spell it out. Combination. It's torn between writing combo and combination. So let's do it this way. It's getting a random combination of genes based on kind of like a random selection of maybe, well, I'm going to pick this one, and I'll pick this one, and I'll pick this one. Okay? I'm going to pick chromosomes from either parent, but we don't know which chromosomes that I'm picking, right? It could be that one, maternal chromosome 1, paternal chromosome 2, paternal chromosome 3, or I might take paternal chromosome 1, maternal chromosome 2, paternal chromosome 2, and maternal chromosome 3. Basically, every time an egg forms, we're getting kind of like a random combination of genes. So it makes sense that the egg that went into sister would be a different combination compared to the egg that went into brother, right? And similarly, the same process occurs with sperm formation. Even though they're both getting a genetic combination from dad, they're not likely getting the same genetic combination because they'd be picking different, completely random combinations from dad's set of dad's diploid genome of chromosomes, right? So, but you kind of already know that because you know that brothers and sisters do not look identical. Even twins don't look identical unless they're identical twins. I mean, if they are identical twins, then yeah, they would have the same genetic instructions because at that point, the zygote had already formed before it split. And once the zygote splits, just like the zygote split, if each of those half zygotes develops into its own baby, they would have to be carrying the same set of genetic instructions, okay? But siblings, apart from identical twins, would have to be carrying different combinations of genes because even though they're coming from different parents. Now, if you take the whole species, if you take the whole sum total of all the organisms in a species, the number of genetic combinations that are there in the different individuals is really pretty huge, right? Okay. So if we have that amount of variation between a brother and sister, even though they're sharing the same mother and father, think about the amount of variation that we see in the whole species, where there's a huge diversity of different mothers and fathers giving rise to all of those individuals, right? And this diversity of gene combinations between individuals is really important because this is what gives rise to to a species, a population's ability to evolve and to adapt to new conditions. Right? Again, think back to the natural selection discussion. We said that, well, yeah, in order for a population to evolve, to become longer armed, right? If longer arms is favored by natural selection, then we need to have variation in arm length in order for natural selection to favor those individuals with longer arms, because we don't have longer arms or shorter arms to choose from, then there's no way for natural selection to work, right? And so, yeah, this type of genetic diversity is what gives rise to the phenotypic diversity that's required by natural selection, and it's going to be inherited because it's underwritten by genes. And then, so yeah, I mean, ultimately what we're talking about is a pretty important thing when it comes to understanding why biology is the way it is, right? And now's the time for us to actually talk about the processes that are involved in meiosis. This is where we're going to be looking a little bit at the textbook, because I kind of avoided that so far. Mostly because the textbook, the diagrams are not all that good. There are a couple that are okay. I'll go ahead and share them with you right now. Right, the first one that is worth sharing, because it's basically telling us the same thing that we were talking about before, is that when you have a diploid organization, right? In other words, this is actually illustrating exactly the same thing that we were talking about before, right? Now, these are two homologous chromosomes. Say they're homologs. They come from different parents. This one might be one from the female parent, maternal chromosome 1. This one might be paternal chromosome 1. They could carry different genes. Okay, so this is illustrating the fact that from the mother had genes that would cause them to be brown coated, like this mouse over here, and black eyes, right? So brown coat and black eyes, that combination was inherited from mom. From dad, we got the combination for a white coat and pink eyes, okay? And so if this is the combination of genes that's in the parent, right? This is the parental genes. Then during meiosis, we're going to be getting kind of like a random pick of either any one of the sister chromatids. These 4 sister chromatids are going to be segregating with equal likelihood. And now, if we only have one chromosome, if we only have one, chromosome 1, then we only will have these 4 different kinds. But if we have 2 chromosomes, right? This is actually, for some reason, they put this in the previous section. Okay. If we have 2 chromosomes, like we're saying here, during the process of meiosis, we're getting a random sample of chromosomes from both parents. Okay, and so what this section of the chapter is telling us is that as things are segregating, as we're ending up with these combinations, we might be getting combinations that are similar to what the parents had, or we might be getting combinations that are actually really different. Right? So again, that's independent assortment. Okay, let's see. Where is part of the chapter that it's in 814? Okay. Again, I had this better in the first version, but since I'm recutting this a couple of times before. No, let me see if I can reorient myself by going back to the original outline. That's the one that made more sense to me. It's a little bigger. Do that. All right. where was I? Meiosis creates haploid sets from diploid parental genome. Okay, that part is, I think, relatively clear, right? Anybody need me to go over that? I mean, did I kind of do that? I said that during meiosis. Darn it, come on, do your thing. During meiosis, we're going from a diploid number of chromosomes, a diploid set of chromosomes to a haploid set. So in other words, your cells contain 46 chromosomes. During meiosis, and the eggs or sperm that you're producing cells with half that number. So yeah, I think it's pretty directly related to what's discussed there. Every combination of genes that's produced by a gamete is unique. Right? That's because we're getting brand new sets of chromosomes. I mean, basically brand new combinations of chromosomes. Crossing over takes place as well. Oh yeah, here's the other thing that is specifically of interest. Okay, there's a real significant difference between mitosis and meiosis, and here's one where I want to go back. This is why I needed that slide before, right? During the process of mitosis, and this is what we did before with our drawing, right? What we have here, each one of these chromosomes is one chromosome, right? This, for example, this might be maternal chromosome 2. This one over here would be paternal chromosome 2. So one chromosome lines up at the metaphase plane, and so we get equal segregation of each chromosome. They go down both directions, right? Now, in contrast to that, if you look over here in meiosis, meiosis starts with 2 chromosomes paired up. Right? So the maternal chromosome 2 and the paternal chromosome 2 pair. In meiosis, and this is the process that gives rise to this is what gives rise to the opportunity for recombination to take place, right? And so we would call this homologous pairing. And it's unique to meiosis, and as a result, we have the opportunity to do a couple of things. We have the process of independent assortment, which basically says we're going to get different combinations of chromosomes, right? The other thing is that we're going to be having crossing over, and maybe you could see that here, or maybe I need to try to make this bigger. See if I can do that. Oh, come on. There we go. Okay, so what we're seeing here is the textbook's representation of recombination that's occurring within a chromosome. Okay, and this is a phenomenon that I'll be illustrating for you. It's called crossing over. And what it allows is for us to get a new combination of genes where you might have maternal chromosome information up until that point. You get paternal chromosome information for the rest of that chromosomal body, right? And so on the other side, we'll have the opposite. We'll have mostly paternal information on this chromosome, but we have maternal chromosome at the very end. That would basically be these 2 chromosomes, these 2 chromatids that have occurred during meiosis, during homologous pairing. They match up, and you have an opportunity for the chromosomes to be cut and then spliced across the homologous chromosomes, in which case you end up with new combinations of genes occurring at a completely different level relative to simply picking different different chromosomes. Okay, homologous pairing occurs in mitosis. Now we're going to be talking about crossing over, resulting in new combinations of maternal and paternal genes within a chromosome, right? So let's say we've got these two chromosomes here, right? This would be a homologous pair of chromosomes, and I'm going to color code them. So one is going to be purple, the other one, I'm going to recolor it. So the maternal chromosome is going to be green. Now remember, this is a phenomenon that only occurs in meiosis. But yeah, for example, during mitosis, you would have spindle fibers coming in over here, spindle fiber coming over here, and so one chromatid would be going to one daughter cell, the other chromatid would be going to the other daughter cell, right? Another spindle fiber would be coming to paternal chromosome 3, and one chromatid would be going to one daughter cell, the other to the other daughter cell. So every daughter cell is going to get exactly one chromatid from each one of these chromosomes, right? Now, what I mean by homologous pairing is that those chromosomes, this is the maternal one, centromere right here, is going to line up and pair up with the paternal one. Okay, so this is the structure that the book identifies as a tetrad. Oops, okay, draw. It's a tetrad. This is a place where you have the opportunity to do crossovers. So a crossover means you're going to be making a cut there and a cut there, right? At exactly the right point, the same point in both of these chromosomes, right? These are non-sister chromosomes, but one chromosome is going to be linked up to the other chromosome like that. The other chromosome is going to be linked up in the opposite direction, right? And when you have a phenomenon like this, we're going to end up with this chromosome, the first one, becoming just the unrecombined chromosome. It looks like that. This chromosome here, the fourth one, is going to look like this with no recombination whatsoever. But now, the second chromosome, this one here, is going to be mostly maternal. But when we get to the end, it's going to be paternal. Back. Come on, change color for me. Okay, paternal there, right? And so what's really neat about this is that this split right here, where one gene, where one chromosome is cut and joined with the other one, that that cutting and splicing is so precise. It could actually occur right in the middle of a gene. And so you might end up with half of an eye color gene from mom, half of an eye color gene from dad, and it's still perfectly functional, right? The cutting and splicing is incredibly precise. And so similarly, when we look at the third chromosome here, this one after the crossing over is going to look mostly like a paternal gene, a paternal chromosome, but the very end of it is going to be maternal. Right? And so, yeah, this is another way, above and beyond mixing and matching different chromosomes. Not only are we getting different combinations of maternal and paternal chromosomes, each chromosome is likely to be a mixture of both maternal and paternal genes. And that further contributes to this idea that each gene combination every combination of genes in every gamete represents a brand new, unique combination of genes, right? It's kind of like snowflakes, right? Maybe it's not entirely true that every snowflake is different from each other, but there's a huge diversity, a mind-bogglingly huge diversity of snowflake structures. And for a human genome, there's an equally mind-boggling diversity of different combinations that are going to occur in gametes. All right? Now, again, why is that important? Well, yeah, right? It's has to do with the genetic diversity, right? If we don't have sexual reproduction, if we only have asexual reproduction, think about it. Every individual produced by asexual reproduction is a genetic clone, right? And so if there's natural selection favoring for longer starfish arms, and all the starfish have been reproduced entirely asexually, then there's no way for the starfish to evolve towards having longer arms because everybody's got exactly the same genetics, right? See the problem there? Now, a different starfish that's reproducing entirely sexually are going to be producing some starfish with long arms, some starfish with shorter arms, because you've got that genetic diversity underlying it. And so that other starfish is going to be able to adapt to the new conditions that require longer arms, and the asexual starfish is going to suffer and maybe go extinct, right? And this is the basic story explaining why it is that sexual reproduction is so prevalent in animals and plants today. Okay? And we'll talk about this a little bit more in a future lecture, but it's a worthwhile thing for us to be talking about right now. All right, that's it for this recut of lecture 11C.